Firstly, a fresh leaf was obtained from kalanchoe plant. Secondly, the two leaf pieces were gently pulled away from one another to peel off the lower epidermis. Thirdly, a small sheet of epidermis were placed on slides. A drop of distilled water and salt solution were added on both epidermis sheet and covered with cover slip. Then both slides were labeled. After that, the slide was placed on the microscope and observed microscopically using low and high magnification power, which are 10 times and 100 times magnification lenses. Immersion oil was used for 100 times magnification lens. From the observation, it can be seen that the stomata closed when the salt solution was added. In contrast, it can be seen that the stomata were open when distilled water was added.
About 450 ml of fascindine were poured into a beaker. 150 ml of fascindine from the beaker were transferred into 3 flasks. About 1 cm was cut from the bottom end of each celery stalk using a razor blade as it remains submerged in water. First celery has no leaf, while second and third celery have leaves. The cut end of each celery stalk was quickly placed into the 3 flasks containing fascindine. Flask A contained a stalk with no leaf, while Flask B and Flask C contained stalk with leaf. Flask A and B were placed under normal laboratory conditions. Flask C was placed directly in front of a running fan. A lamp was also turned on and positioned about 15 cm from the top of the stalk to warm the leaf. All celery stalks were allowed to stand for 45 minutes in their designated environmental conditions. The celery stalks were removed from their flasks after timing. Excess dye was rinsed from the stalk under running water. Celery stalks were placed on tissue paper. A metric ruler and a razor blade was used to cut 1 cm segment from the bottom end of the leafless stalk from Flask A. The cut end of the stalk was examined for the presence of purple dye in the xylem tissue along the outer edge of the stalk. 1 cm segment were cut from the stalk until the dye begins to fade in the xylem. The distance of the dye traveled up the leafless stalk was recorded. Steps were repeated for the remaining 2 celery stalks and the result recorded.
For Flask A, stalk with no leaf in normal condition show the dye travel distance is 2 cm. For Flask B, stalk with leaf in normal condition, the dye travel for 4 cm, while the Flask C, stalk with leaf in front fan and lamp, show the distance dye travel to 5 cm.
By doing this experiment, we can see that collaboration of each member plays an important role in solving problems, especially in experiment part A, where we encounter a problem in finding the clear image of stomata using microscope. Each of us also took turn in preparing the slides for stomata, which helped save more time. Through this science practical experience, we were able to observe the real image of stomata, which have deepened our understanding of stomata structure and function. We also have gained a clearer understanding on how stomata regulate water loss through transpiration by opening and closing its guard cells, and how different environmental conditions affect stomata. By doing this experiment, we can see that collaboration of each member plays an important role in solving problems.
Through this experiment, we learned how teamwork plays a big role in understanding water transport in plants. Each of us had a task, preparing the celery, observing the dye movement, and recording results. And by working together, everything became more efficient. We also realized that doing science is about more than just getting answers. It's about observing carefully and thinking critically about what affects transpiration. This experiment showed us how environmental factors like wind and light truly influence plant processes, and how collaboration makes scientific work more meaningful and enjoyable.